morning, good afternoon, and good evening to all our speakers, chairs, and the wonderful audiences of ACNS webinars in different parts of the world. Welcome back to yet another episode of ACNS webinars. The speaker for the first session of today's webinar is our honored guest from the USA, Professor Babak Khatib. Professor Khatib is the founding chairman of the board of Society of Brain Mapping and Therapeutics and Brain Mapping Foundation. He is also scientific director of the SBMT and Brain Mapping Foundation and director of National Center for Nanobioelectronics, director of Brain Technology and Innovation Park, and CEO of Smart Microscopy Inc. His research has been focused on introduction of advanced diagnostics and therapeutics into clinical neuroscience in order to rapidly identify and introduce game-changing technologies to treat neurological disorders such as brain cancers, Alzheimer's disease, Parkinson's disease, and other brain and spinal disorders. He has been instrumental in collaborating with the state-of-the-art institutions like NASA and bring newer technologies in the treatment of brain disorders. He has won several awards for his outstanding contribution to neurosurgical innovation and education. He is a noted author who has published several manuscripts in various peer-reviewed journals. He is the editor-in-chief of the journal +1 Neuromapping and Therapeutics. We are extremely honored to have him today as a speaker at our webinars, and today he'll be talking about his favorite topic: neurotech, nano-neurosurgery, NeuroAI, predictive modeling, neurophotonics, and mixed reality. The speaker for the second session of today is our honored guest from China, Professor Qian Rongjun. Professor Rongjun is the Chief Physician, Department of Neurosurgery, Henan People's Provincial Hospital, China. His clinical interests are focused upon cerebrovascular surgery. He is a standing committee member of several prominent organizations in China like Trauma Society of Henan Medical Association, Neurosurgery and Cerebrovascular Professional Committee of the Henan Medical Science Popularization Society, the Neuro-Oncology Group of the Henan Medical Association, and many more. He is a noted author with several publications in various peer-reviewed journals. We are extremely thankful to him for accepting our invitation to be a speaker at our webinars, and today we'll be talking about characteristics and surgery of juvenile arteriovenous malformations. The chair for the first session of today is our distinguished faculty from USA, Professor Walter Sheen. Professor Sheen is the Chief of Neurosurgery at the Lehigh Valley Health Network, Pennsylvania. He is also the professor at the Department of Neurosurgery at the Morsani College of Medicine, University of South Florida, and clinical professor at the Department of Neurosurgery at the George Washington University School of Medicine and Health Sciences. Professor Sheen is the recipient of several awards and grants for his outstanding contribution to neurosurgery. His academic excellence is evident from the scores of publications that he has published over the years in various peer-reviewed journals. Professor Sheen has kept his commitment to teaching of young neurosurgeons globally with his online monthly lectures that he has named the Surgeon's Log. He is also the founder and chief executive officer of the Global Brain Surgery Initiative, which is a global neurosurgery educational organization. We are extremely grateful to him for accepting an invitation to chair the session of Professor Babak Khatib. The chair for the second session of today is our honored guest from Czech Republic, Professor Vladimir Benes. Professor Benes has held the position of head of neurosurgery in Central Military Hospital, Prague, and is also chairman of Department of Neurosurgery in the First Medical Faculty, Charles University. He was the past vice president of the European Association of Neurosurgical Societies and chairman of the training committee of the ENS as well as the president of ENS. He headed the neuroanatomy committee of the WFNS along with Professor Imad Kannan in the past. He is an invited faculty to various workshops and conferences organized all around the world, and his literary contributions sums up in scores of published articles in various international peer-reviewed journals and book chapters. We are extremely grateful to him for accepting our invitation to chair the session of Professor Qian Rongjun. On behalf of the Education Committee of the ACNS and the President, Professor Yoko Kato, I would like to welcome both the speakers and chairs and the wonderful audiences to this online platform of ACNS webinars. A very warm welcome to our colleagues in China, and we are extremely thankful to Professor Shubin for broadcasting this webinar on the WeChat channel. Dr. Liu Bun Singh from Malaysia is my co-host for today, and with that introduction, I would like to hand over this podium to our first chair, Professor Walter Jing.
Thank you, Raja. Again, no one does introductions better than you across the world of neurosurgery, and I'm very grateful for this opportunity. So about technology, our field is driven by technology, and every about 2 decades or so, neurosurgery takes a giant leap forward from the introduction of the microscope, the next phase being the introduction of the CT scan and MRI machines, and now we're talking about artificial intelligence, machine learning, mixed reality, and all that. Our speaker today is a leader in this technological-driven field and discovery of new innovations and new ways of using technology, Dr. Babak Khatib. And as Raja told you, he is the brain mapping is more than just brain imaging. He's going to tell you about that. We're also going to learn about nanotechnology and how he uses mixed reality in neurosurgical advancement. So my main job right now is to get out of his way and let him speak, and then we'll have some comments at the end.
Professor, thank you very much. I also thank you, my colleague, Professor Raj, for this fantastic opportunity and the organizing committee. It's an honor to be here, and I'm humbled to be selected by your committee to give a lecture on the Neurotech, which is basically really expanded to a variety of new platform. Let me share my slide here. I have 42 slides, but I will go very fast. And all right, let me just share the screen here. So today we're going to talk about neurotech, nano-neurosurgery, AI machine learning, neurophotonics, and brain mapping, and cellular therapeutics. As Professor mentioned, I have several different hats as a chairman of the board of Society for Brain Mapping and Therapeutics and the Brain Mapping Foundation, but also director of National Center for Nanobioelectronics and Brain Technology Innovation Park, which is a $2.5 billion initiative with Biden administration. I was also advisor to President Obama for the United States Brain Initiative and instrumental in other international initiatives on brain. So I'm just disclosing here that I'm chairman of the board and the CEO of Smart Microscopy. This is a spin-off company, came out of NASA. I was a NASA scientist until 2020. And one of the technologies that we use is a multispectral imaging to be able to detect tumors intraoperatively. So you're all neurosurgeons and familiar with intraoperative mapping, but I'm just going to show you a very brief video here. Let me, prior to do that, let me just unshare and share to make sure that I have a voice on the videos. I'm sorry, 1 second. Okay. It's a skull-based procedure that you're all familiar.
So that's the.
And we're trying to track the tumor.
There is nothing else. This is a great tumor. I mean, there's nothing else. We are looking backwards and the supramedullary is right here.
In this particular surgery,
Exactly that.
The navigation system is off, the virtual reality system
This is the other side, this is the left side.
End up
The right side. Okay.
Based on the anatomical location.
Going a little bit more.
Tissue at the tumor. In the first biopsy on the lung.
Okay.
Later on,
I think I'm in the right spot.
Was a tumor. So the point being is that there is a need for real-time intraoperative navigations that are optical based. So in this lecture, that I have 5 objectives to share with you. First of all, introduce what the Brain Mapping Foundation is, what's Society for Brain Mapping and Therapeutic, what's the Brain Technology Innovation Park, and what is brain mapping in general, and what are the examples of it, technologies that can help applications at the outcome of new emerging field of stem cell therapy, and also what should be the future directions of research and development as well as funding for this field. Okay, I'm just gonna say that the mission of the Society for Brain Mapping and Therapeutics is basically improving diagnostics and treatment and rehabilitation of patients afflicted with neurological disorders. Of course, we do this in a multidisciplinary approach, and we bring scientists of different backgrounds, surgeons and NASA scientists, electrical engineers, software engineers from Microsoft and Intel, and basically allow exchange of ideas. So this is a short video for you to enjoy.
For brain mapping, does a fantastic job bringing together experts from multiple fields.
From geneticists to engineers to neurologists.
You know, we've got rocket scientists here, we've got neurosurgeons here, all thinking about how to solve very difficult problems.
What this organization represents is a leading edge integrator for the disciplines that exist in medicine wherever they are, and actually in engineering.
The challenge is making sure that we're able to cut across all of these silos to make sure that we're sharing the information and providing collective power of our new knowledge. And I think SBMT is clearly representing that venue, that platform that allows that to happen.
The efforts in the background of this organization will assist all of us who work in neuroscience.
The organisation is a leader in terms of advocacy in bringing key people together, both academics but also key policy decision makers.
We develop a lot of technology for space exploration at Juvenile, but there's lots of application of these technologies in the medical field. And of course, being part of the society puts us directly in contact with these folks and helps us tremendously to get those technologies transferred out into the medical industry.
What is talked about in terms of treatment and diagnosis here is really going to be the forefront in the future.
At a minimum, be engaged. Share your stories every chance you get. And don't be shy about expressing your needs and concerns. Nobody else is. How we make decisions, communicate, and process information is a vital part of this new frontier.
So you have a good understanding of, I'm sorry, next slide, I would say. And okay, so that takes us to definition of brain mapping. As Professor Walter Jean mentioned, we have redefined as an organization the brain mapping from brain imaging to the study of anatomy and function of brain and spinal cord through the use of imaging. So that use of imaging could be multi-modality imaging, immunohistochemistry, and all the way to optogenetics and cellular, biological, and nanoscale. So, and then we have published the inaugural textbook of NanoNeurosurgery and NanoNeuroScience. Within that framework, we define the application of nanotechnology in neuroscience as nanoneuroscience, and the therapeutic application of nanotechnology, which could enhance neurosurgical treatment, is called nanoneurosurgery, and integration of nanotechnology with device, cellular therapy, and imaging called a nanobioelectronic. And I will give you some example of that. So we do have annual meeting like Congress of Neurological Surgeons and American Association of Neurological Surgeons. So Society for Brain Mapping has held 19 World Congresses, and the next one is our 20th, which is the largest convention ever, with 1,500 speakers. Will be next year in Los Angeles. Out of these conventions, obviously knowledge is created. And we have published two textbooks of inaugural textbook. One is NanoNeuroscience and NanoNeurosurgery. When I was advising President Obama, I basically signed a copy of that for him, and he thanked me for my contribution to the science. But also the next one is the neurophotonics and brain mapping. These are the inaugural textbooks that are basically bringing optics and multimodality spectroscopy to intraoperative mapping, and they're both actually up for new edition, and we're working on both of them, publishing with Nature. As I mentioned, I was one of the advisors to President Obama, for the Brain Initiative. I was at the White House when he announced it. But we were very active on the policy side. I see one of our colleagues from Malaysia here at the corner here on the bottom. You see, I gave a keynote to the Malaysian Neurosurgical Association back in 2014 or 15. And we actually started collaborating with the Malaysian colleagues to build the Malaysian Brain Initiative. but also spoke at the Parliament of Australia and the Parliament of Canada. Basically, we launched the Canadian and Australian Brain Initiative. We have been advocating in United Nations, talking to the head of NASA or Supreme Court Justice Elena Kagan about the importance of basically setting up science policy or policies that could actually help fast-tracking therapeutics. Here we are, we awarded Stephen Hawkins for his dedication to raising awareness about ALS. And we have been working with Office of Science Technology Policy at the White House, and NASA JPL, and the Congressional Neuroscience Caucus, which is bipartisan, congressional, both Republican and Democrats. We also launched the IEEE Brain project. At the G20 Summit in 2014, we started what's called Neuroscience 20, that's brain, spine, and mental health. We looked at the cost of neurological disorders to the world economy. Actually, it's staggering. It's about $16 trillion, and that was prior to pandemic. And we have been advocating and educating policymakers. Here we are with the President of the European Union, previous President of the European Union, and we have held this summits in parallel with the G20 summits in the last several years. So we published 2 papers on this topic, looking at the cost of neurological disorders, but also basically providing some solution and ideas. One of them was formulation of national data repository and analysis for neuroscience, which we are working with NIH to establish that. The other one, creation of national network of brain procurement, allocation, and banking, which basically provides the viable and processed tissue to all scientists to have an access to basically human brain for studies. And the third one is establishment of Global Alliance for Nanobioelectronics in brain mapping and therapeutics, which basically allow us, all of us from our colleagues in Asia and Europe and so on, to interact and share data and collaborate. And finally, facilitate rapid introduction of diagnostics and therapeutics, which basically going to, as I said, impact the financial and the human cost of neurological disorders. I mean, this is basically a chart that shows you the cost of neurological disorders in each category, which was published in our paper in the Journal of Alzheimer's Disease. As I mentioned, I'm a director of a Brain Technology Innovation Park. This is a $2.5 billion initiative with the Biden administration and California governor to bring to basically build a biotech park for brain, and we are actually submitting several legislation with the United States Congress and Senate to fund this effort. I hope that this video will play here.
We have an initiative called Brain Technology Innovation Park. We are going to construct a new biotechnology park in Los Angeles.
Brain Technology Innovation Park is a special type of incubator. What an incubator does is it takes ideas and it brings them to market faster and at lower cost.
Obviously, it's a very large investment. It would need tremendous support both from the state of California and from the federal government. I think it would be a game change to try to really develop that kind of a multidisciplinary neuroscientific brain power consortium right here in California.
Certainly the Brain Technology and Innovation Park is something that's going to foster and forward science in general. It's really several $billion dedicated to neuroscience. That's going to be inclusive of putting money towards things like Alzheimer's, multiple sclerosis, Parkinson's disease, and also stem cell research. Those are well-needed things in the complexity of what we're trying to fight, particularly in those neurodegenerative diseases.
So what I like to see is to see more successful story of translating all this discovery into actual clinical setting, and then try to use this method or discovery into actual practice.
Why neurotech? Because neurotechnology is an emerging field, like AI. Years ago, you would hear about augmented reality and virtual reality. That was a great story about new advancement in science. Now you're hearing about AI, machine learning, and supercomputing. But the next generation of technology will be neurotech. Your car will be working based on your thoughts. The technologies that we'll be having is going to be basically work off of your neuronal signaling. Neurotechnology is a great field that's going to be revolutionizing our living.
Yeah, so as you know, Elon Musk just put about $500 million in Neuralink. So there will be a lot of technologies in this field. So the way that we have set up the foundation and the society and our international N20 initiative is to basically create a global network of multidisciplinary scientists through association as members, but then provide funding to their research through the foundation, and basically have a consortium of different brain initiatives that we are coordinating or collaborating with through the N20 and push policies forward in that regard. So we have created this network of government agencies with Department of Defense and NASA and national labs, also with the societies and associations, and we welcome you to be one of our collaborators, and also with universities and entrepreneurs, hedge fund, investment funds, and also kind of a service providers that basically push this technologies forward. So I'm going to dive deeper into this topic of technologies that we will be advocating and how these technologies actually will help neurosurgeons and clinical neuroscientists to advance therapeutics. So with that, I'm going to go over basics of the physics of waves here. So you're familiar with X-ray, and in this case, 0.1 to 10 nanometers. UV spectroscopy is 10 to 400 nanometers. I want you to kind of take a note on that. And infrared is 700 nanometer to a millimeter, and microwave is 1 millimeter to 1 meter. So here is one of the examples of how we actually used a microwave technology to develop a microwave ablation for treatment of cancers of the brain, prostate, and liver, and so on, to basically address the clinical needs of it, including metastases. So this is a FDA-approved technology. All right, this is a device, microwave ablation device that we have invented in the Department of Neurosurgery at Cedars-Sinai, that is a microwave with a chokehold, and can create actually a lesion for the size that we want. The device is actually controlled by the computer, any laptop, and the plan is that at the time of a biopsy, we basically put the biopsy needle out and put the microwave ablation in, and basically ablate the brain tumor. So the ablation in this case is a microwave. So as you know, the way that microwave works in your microwave oven, it basically vibrates the water molecules, and that water molecules heat up the tissue and the nature of the protein. So the basics, the basics of physics of this is as such that you have a microwave with a chokehold. So basically, you don't, heat up the whole brain. You just dial the size of this, lesion that you want to make based on preoperative MR imaging. So you basically cook the brain tumor. It's a better technology than currently used, let's say, radiation technology, which has radiation necrosis and a variety of other problems with it. All right. So diving deeper into nanomedicine and emerging technologies, this is NASA's carbon nanotubes, and we actually use carbon nanotubes in this case. As you know, nanotubes are, carbon nanotubes are 200 times harder than steel. So how do you actually use them for drug delivery? In this case, we actually took carbon-carbon bonds, put sulfur at the middle. So basically, a biological a tissue or cell can actually oxidize sulfur and break down the carbon. We also could use this as siRNA and cytokine delivery system, and tag them with a poronic dye so you can actually detect them. So in this case, we published a paper showing that we could use these functionalized carbon nanotubes and have cytokines attached to them, which could be picked up by the microglia, which could activate the microglia to attack brain cancer. So that's this is a human microglia with carbon nanotubes in them, and it's a short video that you could actually see. And poronic dyes basically make them autofluoresce. So the nanotechnology of today actually will pave the future for the personalized nanomedicine. And you have most probably experienced this yourself because of the Pfizer and Moderna vaccines. There are nano platforms that allow actually vaccine to be more effective. Well, this is the same carbon nanotubes application in vitro. That was in vivo. This is in vitro. And in this case, you can see that the macrophages are in green color and the carbon nanotubes are in red. And the tumors are in blue, and basically you can see that macrophages are actually traveling to the tumor area. But also we looked at the cellular cytokine profile. So if you look at the previous slides, you could see that the cells are tolerating this functionalized nanotube. The cytokine profile goes up, but then kind of plateau. So to just give you a couple of points on nanotechnology, the importance of nanodrugs are their ability to truly bypass the cell membrane and be very effective very quickly. But also you have to make sure that these platforms, they don't attach to each other, which could actually cause problems in the liver and kidneys. So we also use them for imaging purposes. So this is another paper was published in the scientific report of Nature. We use basically AC magnetic field, stimulated nanoelectroporations of the magnetic, electric, and nanodrug carriers inside the CNS to be able to actually image cells. So in this case, we looked at the cells inside the mice, and we actually looked at the cells injected in the primates. So, I mean, one of the most important aspects of nanomedicine is cellular imaging. And why is that important? Because you have heard about stem cell therapy, immunotherapy, cellular therapy. You really want to make sure if you're putting 10 million cells in someone's body, you want to see where are they going. And always FDA going to ask you, okay, you're putting X amount of cells in someone's body. How do you monitor them? Well, that's how you monitor them. You attack them with the nanoparticles. Another platform that we've been working on is they're called NanoDrugs. These are nanostructures that filled with drugs. So in this case, you have a nanostructure of a TEG, and in this case, you put ibuprofen and alpha tocopherol. And ibuprofen is, as you know, vitamin A, and alpha tocopherol could be also bind to the octadecanethiol, so forming this kind of a nanoprodrug that could be also tagged with cis5 or gadolinium, which could actually help you make drug delivery in the case of, let's say, traumatic brain injury. The new generation of drugs for traumatic brain injury will be nanodrugs, which you could actually inject at the time of injury in the battlefield or car accident as a paramedics, which could go to the area of injury, reduce the inflammation, increase oxygenation. So again, this could be used for drug delivery to Alzheimer's, stroke, and for chemotherapy of the future. Nano platforms are also used as nano films. In this case, NASA used their nano films to trap compounds in the International Space Center. So in the International Space Center, they're in a closed kind of area. They want to make sure the toxic gases will not harm our astronauts and cosmonauts. Therefore, they have this, electronic nose that sniffs the air quality. And the way it works is that basically they're nanofibers, they trap the compounds, toxic compounds, and basically they give you a signal. Well, in this case, we say, okay, why don't we actually smell or test this on brain cancer and other tissues? And we basically discovered that actually brain cancer, they have a different signal on this electronic nose. And so basically they published this in NeuroImage, and they received an award from NASA. Another technologies that we have transferred from NASA to neurosurgery are multispectral imaging, including infrared imaging. So in this case, we use NASA's infrared to detect metastatic melanoma. We're all familiar with this. I'm just going to run this, two videos at the same time that you could actually see intraoperative infrared thermography for tumor delineation, which I'll show you later on how we have used this for basically predictive modeling and therapeutics. In this case, you see that, as you are all aware, metastatic tumors are much more rounder, much more vascularized versus gliomas and GBM. But there's a great value in real-time multispectral imaging, because you can't really use MRI in real time. There were some efforts to use double donut MRIs in the past, as you're all aware, but those really failed. So in this case, in the video at the bottom, you see the tumor in the petri dish sitting still, having a different temperature, higher temperature. And that speaks to the fact that these tumors are very active metabolically. And you can see still there's some residuals of this tumor left, which was confirmed later on by imaging studies in this patient. This was published in NeuroImage, and basically we're now working on new generation of multispectral imaging with NASA. And of course, you're all familiar with surgical theater, and that's the augmented reality. I'm sorry, the virtual reality. Sorry that I forgot to add our augmented reality video here, but we're now working on augmented reality with Microsoft, which is much more effective system. But as you can see, we could augment these images, and I'm sure many of you have used augmented reality in your surgical practice, and could not only orient yourself better, but also educate your patients and students better, as well as getting better outcome. So these are new neurotech technologies that are coming around, and some of them you're familiar with. Well, as I said, we are working on integration of technologies with different platforms, and in this case, we published another paper looking at fluorophores in brain cancers. So, as I mentioned earlier on in my talk, we use basically physical wave, I would say, properties of fluorophores to detect them. So what are the fluorophores? Fluorophores like NADH. You're familiar with NADH or FMN. These are compounds that are in the Krebs cycle used, but they're actually upregulated in brain cancers, and they have optical signature. So based on those upregulations, you can look at the peak excitation of these fluorophores and actually use them for intraoperative mapping. So in this case, we actually use them to detect where the normal cells are. In this case, the blue dots are normal cells, and the yellow dots are all cancer cells. So that's called multispectral imaging. Okay. Well, of course, we are all familiar with the AI, and you have heard about them, and everybody's talking about AI, and in this case, we are collaborating with this AI agnostic company. But really, the job of AI is as good as data that you provide to it. Okay, so if you provide a lot of nonsensical data, you're going to get a nonsensical kind of outcome. So it's very important how you provide this data, and of course this AI has been used for tumor, dementia, stroke, and a variety of different platforms by integrating different data sets. Well, in this case, we have integrated multimodality imaging, and kind of use the AI machine learning for better tumor detection, and of course using the diffusion tensor imaging tractography for the case of Alzheimer and dementia. All right, so that's great. So we know the optical signatures of fluorophores, and we have the data set that we could gather from these patients. Okay, how do we actually apply that in glioblastoma? So in this case, we're all familiar with 5-ALA that's been used, but multispectral imaging is actually much more effective than 5-ALA. So, so you're pretty much familiar with this, kind of using optical spectroscopy for detecting brain tumors on the left side of the screen. But once you have the data and AI machine learning, you can actually really do a predictive modeling, some cool stuff here. So you can actually predict the pattern of growth of the tumor. Therefore, from one side, you can actually debulk the tumor and make sure that the tumor is taken out completely. But from the other side, you could actually predict the growth of the tumor, which could help you for adjunctive therapy, could help you with all types of planning ahead. We could also use, in this case, we published this paper looking at the visualization method of the spatial distribution of neural stem cells in the gliomas. As you may know, stem cells seek gliomas, basically, cancer. And for a long time, they have been used as a delivery vehicle for drug delivery to brain cancers. So in this case, we actually try to use kind of a mathematical model looking at the distributions of stem cells in the glioma, and you could see, kind of the red dots are just, cells that are scattered through the throughout the tumor. So, in conclusion, I just want to mention that integration of cell therapy or cellular therapy with nanotechnology, device, AI, and brain mapping is the really key to the future of the field of medicine in general, of course, in neurosurgery in particular. How do we acquire data, as I mentioned? How do we share it, store it, and use it? Is going to impact AI reliability. But integration of such data with cellular therapeutics will impact the stem cell therapies of the future or cellular therapy of the future. And I also encourage that scientists should participate in national debates on science and technology, educate policymakers about science and technology. And integration of nanotechnology, AI, VR, mixed reality, supercomputing, and multimodality brain mapping, imaging and cellular therapeutics, neurophotonics, and other devices will be the future of brain mapping and therapeutics. Of course, none of this can happen in a vacuum. So discovery, innovation, and pioneering works is only possible through collaboration. Therefore, I welcome you to collaborate with us in the Society for Brain Mapping and Therapeutics and be part of our global consortium for nanobioelectronics and brain technology innovation part. With that, I thank my colleagues, and I would like to appreciate my collaborators, which includes Lawrence Livermore National Lab, Oak Ridge National Lab, IBM Watson, NASA, and NanoSx, but also some of them are listed here, including Microsoft and Intel. Thank you very much, and I will take any questions you may have.
I am stunned by that lecture, and I have lots of questions for Babak. My first thought is that there's a lot of things going on that you talked about, from brain tumors to AI to do you see yourself as an integrator for all these different sort of aspects of research? One person can not be involved in all these things and be heading all these efforts, right? So can you describe what you see your own personal role in this is, and how you think that all these different branches are going to come together in the near future?
Sure. I mean, obviously, this is a collective work of different groups that we have, kind of organized, which are presented. But all of these technologies are published, so be happy to provide the papers to the committee. I think, as you mentioned, Professor Jean, the integration is the key. That is really kind of a emerging field of neurotech. So for a long time, we think neurosurgery is kind of a different field, and neurology and neuroscience kind of thought to be much more basic neuroscience. But the integration of these fields and other fields, including nanomedicine, AI, machine learning, computer science, really is going to drive the future of neurosurgery. So you will see a lot of disruptive technologies, including what I did not discuss here. New generation of supercomputers are called neuromorphic computing. I don't know if you're familiar with neuromorphic computing, but the quantum computing is basically stuck at this point. And the companies like Microsoft and IBM, they're going to actually use neurons to run the calculations. That's faster. So there is a device, I mean, there's a computer neuron interface. It's called neuromorphic computing. So imagine these type of technologies coming to neurosurgery. It's completely revolutionized the way that we do procedures. And then you will see, of course, a new generation of microscopes, which I would say microscopes are things of the past. I would say exoscopes and augmented realities or mixed realities going to be driving the intraoperative navigation. And then on the treatment sciences, on the therapeutic side, you see more and more cellular therapy becomes, prevalent and becomes much more relevant. In the state of California, we just passed Proposition 14. We have put $5.5 billion aside for stem cell therapy. So, and those of you who are interested in collaborating, there's a significant amount of funding available, and $1.5 billion of that is actually for neuroscience. So there are, as you can see, there's an integration of not only science and technology, but policy. That's what I want to really bring this to your attention. It doesn't matter if you're in China. I was in China and during the G20 summit, we would talk to the Chinese policymakers, or in Germany or Argentina. We need to educate our policymakers. We need to bring them to our labs for them to see what they're doing. For instance, I hope that you know who is your local congressman is. So it's very important for them to see, because then they could put policies in place that could help delivery of care.
A couple of comments. Interesting that you mentioned the exoscope, because Vladimir is a big exoscope user and he's right after me. While I was listening to your lecture, I cannot but recall a very interesting comedic act that said that, we have Amazon Prime, 1-day delivery. Oh, that's not going to be good enough. The future will be you read my mind as to what I want to buy, and you make it materialize in my hand the moment I think of buying that thing. So that was what you were talking about, neural computing. And I fully believe that you're right in that treating glioblastoma there's going to be no knives anymore in the future. A microscope, forget about the scalpel. future of neurosurgery is going to be no knives at all. I think we're going to look back on cutting out things as barbaric very soon. But when is that happening? When do you see the next big quantum leap happening with molecular medicine and all this?
Well, great question. To give you another example, you're familiar with the coiling, right? So we use coil for basically aneurysms. So new platforms are coming up in nanomedicine that completely bypass coiling. Basically, you inject the nanoparticles, they completely seal it. Okay, so I mean, I think what we are trying to do with the Brain Technology Innovation Park initiative is to really fast track those technologies that are already existing in other fields and bring them to clinical practice. And the NIH does not fund that, what we call value of death, okay? So if we provide the funding, you fast track that therapeutics, and then basically we create an operation warp speed for neurological disorders. So when that's gonna happen, I hope sooner than later, but I would say probably within the next 5 years, you're gonna see complete change in neurosurgery. You will see a lot of cellular therapeutics and a lot of cellular byproduct therapeutics for ALS, for MS, and for brain cancer. You know, I spent 20 years of my life in studying of brain cancer and going after treatment. We really have to go back to the basics, okay? And the basics is that for all of us, we're taught in medical school that there is no lymphatic system in the brain, right? Oh, wrong. We just discovered that there is a lymphatic system in the brain. Well, if there is a lymphatic system in the brain, then you're going to be in a situation that brain inflammation causes a variety of different disorders. Now we are seeing that you know, brain inflammation could be the cause of Alzheimer, cause of ALS and MS. So that brings us to the brain health. Again, that brings us back to the policy. We just published a paper in Journal of Alzheimer's Disease saying that why don't we have a brain screening, for God's sake? You have a screening for prostate, for breast, and you know, lung and so on. As part of the healthcare, we need to have a brain screening. So we put together a paper and published that in Journal of Alzheimer's Disease. So the policy goes hand to hand with science and technology, right? So and if the policies are shaped properly, the science and technology gets faster to the masses.
Raja, any questions from the audience?
Yes, of course. Thank you very much. First of all, I would like to thank Professor Babak Khattab for this mind-boggling lecture. Much of things went above our heads, but we understand the future is bright and complex as well. We can take a few comments from our guest. Professor Benes is here. Professor Benes, any comments from you?
It was amazing, you know, one sees the future, but somehow, if I understand properly, we shall be performing less surgeries, we shall be more gentle with the patients, and neurosurgery per se will, as you believe, change dramatically. And we are going to be more or less like neurologists, as we are now also, obviously. Am I right?
Well, I would say you will be much more neurotechnologists. So I think for a long period of time, and neurosurgeons were disrupting neurosurgery. But now you see Elon Musk come and put $500 million in neurosurgery, actually. He created a company called Neuralink. So you will see a lot more entrepreneurs coming to neuro. And that's why you will see a neurotech. You have heard about biotech, you have heard about, tech in general, but neurotech is in, kind of a, how can I say it? It's a fast advancing field. It's not only in neurosurgery though. You see in US military, when you are basically taking command of a supersonic jet, right? If every millisecond of reaction counts, right? So the new generation of fighter jets actually works with your brain. So brain-computer interface, the helmet that they have basically can control the jets. So, I mean, that's just on the military side. On the, on the therapeutic side of things, you see much more non-invasive neuromodulation. So right now, of course, you're familiar with deep brain stimulators, all types of stimulators for, vagus nerve, for epilepsy and so on, but those are invasive. Those requires neurosurgeons. But what if I give you a kind of a sensor that you can put it, like a, little bandage or whatever you can call it, a strip of sensor, you can put it on your head and stimulates your brain. You don't need integration. And those are not that far away. I mean, this is not like, this is not science fiction. There are people working on this type of technologies. So there are people working on really reducing the size of surgical technologies. You remember the double donut MRIs that they were huge, like a Russian tanks, and then you had to build the entire building around them, and 40 tons of MRI machine. But with mixed reality and optical spectroscopy, you don't even need that. So there are disruptive technologies coming, and our job as neurosurgeons and neuroscientists is to really embrace them and kind of allow them to be integrated.
That's perfectly true, but somehow I have the feeling that there is some biological ceiling. We hit it with neurotransplantation, we hit it with neuroprosthesis, we even hit it with neuromodulation. That it really depends on technology, I believe it, but somehow all these newest things hit the ceiling, that we were unable to overcome the problems with neurotransplantation, which is not really in clinical use. The neuroprosthesis, there are let's say, hearing implants were abandoned. There was a big hit some 15 years ago, and somehow it's over. The cameras for blind people with stimulation of the visual cortex, it was a hit. I have seen it for the first time in 1985, and I have seen the same patients in 2020. What do you think about this?
Yeah, I remember the creator of that camera, actually. Well, the whole system was done by my friend Marco Mayon in collaboration with National Labs. So one of the reasons that the implants fail is that when you're inserting an electrode to a brain, it causes damage, right? And that damage creates a process called gliosis, basically cements the surrounding of that damaged area, so it does not allow the electrode to pick up the signal. So those are the issues that are now addressed. Actually, I have a patent at Caltech that at the time of electrode insertion to the brain, if you're injecting cytokine, actually it blocks that gliosis process, therefore extends the life of their prosthetics. So, I mean, there are basic issues, absolutely. It hits the ceiling until there's a solution for it. So that's basically where we are. But I mean, in terms of the future direction of neurosurgery and what's coming at us as a scientist and surgeons, it's absolutely bright. But in a sense, it's much more precision-guided medicine. So the therapy that could be used for me will not be used for you, because the way it's going to work is that, you know, we're going to take a piece of our skin and develop an iPS cell, right? Basically stem cell, test the drug on the stem cell of this person. You know, basically, after that, you have a dose response for this person and basically personalize the medicine for this particular individual. So that's kind of where cellular therapy, not only on the diagnostics and therapeutics come to play, but also even in the prevention areas. How do you prevent people getting Alzheimer's? How do we prevent in the future? How do we prevent people get brain cancer? So,
thank you. Thank you. It's
Thank
great
you very
vision.
much.
it's an
Extraordinary.
honor to be here. Thank you.
It is extraordinary. Thank you very much. So we have concluded the first session, and I would like to inform before that, I will of course take final remarks from Professor Walter Jean. I would like to mention
my only
Yes.
final remark is that I want to thank Babak for that lecture. That was really mind-boggling and mind-opening as well. Vladimir and I were together face-to-face just last week, and I want to tell him that I just booked my trip to Prague. So there you go, Vladimir, to you.
Thank you. Great.
I hope I see you all in our next World Congress, and we should not end our collaboration here. We should expand it. I extend my collaborative hand to you. As I said, Professor Jean and colleagues, it's truly an honor to be here, but let's collaborate on exciting projects.
Thank you. Thank you very much. I'd like to mention to our viewers that this is being broadcasted on WeChat, YouTube, and Zoom. And as of now, we have 1,225 people who have joined us live from all across the world. And we're extremely grateful to Professor Shubin for broadcasting this on the WeChat channel. So I would like to thank Professor Babak Katib and Professor Walter Jean for taking out that time. Thank you very much, Professor Babak. Extremely sorry to wake you up so early in the morning, but nevertheless, it was a great lecture. Thank you very much.
Best way of starting a morning.
Yes, of course. So we'll move on to our second session, and I would like to invite Professor Vladimir Benes for the introduction part.
Okay, thank you, Raja. We now have more clinical lecture, I guess, by Professor Rongjun Xian. I'm sorry if I do not pronounce your name properly, but I really do not know how to read it. Professor Xian is from Henan, which is one of the important parts of China. He now is the chief physician of neurosurgery in Henan Provincial People's Hospital. However, he started his education in Guangzhou, where he got his doctorate in surgery. And then he worked for a while in Germany, where he was in a trauma center, one of the largest one in Europe. And he also worked in Ethiopia in Beijing Hospital. Must have been fun for you, I guess. His academic activities are very broad and very, let's say, the articles are from various fields, starting with nanotechnologies, so something which would continue on the first lecture. But later on, he got into gliomas, which were his, let's say, main interest. And if I understand the publication and its sequence, Professor Xian is slowly diverting for vascular. And vascular will be the next lecture. He really did a lot of work in imaging. He did a lot of work in NanoDrugs. He did a lot of work in sphenoid rhythm meningiomatry, which he had a project of Henan Technology Research Institute. And he was also interested in individualized treatment of craniocervical junction malformations, which is a very interesting field. However, today, Professor Xian is going to talk on juvenile arteriovenous malformations. So let's give space to Professor Xian and let's listen to the lecture. Professor?
Okay, thank you, Professor Benes. Yes, just know the professor cut the speech is very good, is wonderful, and some things is amazing me. So I think I know I have just some things to learn from him. Thank you. Okay. Now next times and I will give speeches about the characteristics and surgical treatment of the malformation, the cerebrovascular. So as we know, and I come from the Department of Neurosurgery, Henan Provincial People's Hospital, China. Other names is the Zhengzhou University People's Hospital. Today, my speech is the characteristic and surgical treatment of the juvenile cerebral vascular malformation. Because in our province, we have the big problems here. And so, and in ours, the clinic works, and we can always see the juveniles, the cerebral vascular malformation. First of all, let we see the abdominal region of the AVM. Cerebral arteriovenous malformation is a leak of capillaries between the cerebral arteries and the cerebral veins at the lesion site, resulting in the direct communication between the arteries and the veins, forming a short circuit between the arteries and the veins, resulting in a series of cerebral hemodynamic disorders and corresponding clinical symptoms. Its annual incidence is about 1.3 cases per 100,000 patients. The age of the onset is more than common in 20 to 40 years old, and intracranial hemorrhage occurs in about 50% of the patients. The remaining symptoms include sinus, headache, focal neurological defect, or asthmatomitis. Yes, the symptoms is normal and shows the location formation of the ends. The frontals, the deep, paratals, occipital, and preventricular, and the temporals, and brainstem, the cerebellars, and as well. As we know, the special margin gradient is normal in the clinic. And from this gradient, we can see there's those the location located in the function areas. Examples is the centers, monitors, cortex, language errors, the visual cortex, hypothalamus, the internal capsules, brainstem, cerebral, the pendular, and the deep cerebral nucleus were scored 1 point. Otherwise, it was listed as the quit zones, and the score was 0 points. Some of the genes, the variants that lead into the deep vein were scored 1 point. Otherwise, the 0 point. Other raw venous, the malformations, the diameter, maybe the diameter less than 3 centimeter is 1 point, and 3 to 6 centimeter is 2 points. If the more than 6 centimeters is 3 points. The total sequence obtained by adding the above the 3 points is the grade of AVMs, which has a total of 5. We can using this the gradient to class the AVM. Oh, this is the observed formation like this. But how treatment of this then? So is this the control with the AVMs treatment? invasive and invasive interventions. Invasive interventions include microsurgery, steroid tactics, the radiotherapies, and endovascular intervention. The management of the unruptured immunity has a history to be the controversial. The 2040, the Arabers, the trials, compares the outcome of the interventional and conservative treatment and conclude that the natural history of the unruptured AVMs was superior to the outcomes of any treatment models during relatively short following the period. But this the following up the period is only the 33 months. After AVMs, the invasions, the interventions are preferred to prevent the risk of the reblating. How to choose the treatment strategies as like this? As we know, the generous the AVMs like this the characteristic. Maybe the more common the intenders, bleeding are more common symptoms. Acute onset, the symptoms of the high intracranial pressure. If it is not in the functions area, the prognosis is better. And even if there is a functional disability, the general recovery status is accepted from the after the surgeries and others the interventions treatment. So next, and I will show the some case and to these audiences how to get the treatment of this patient. The case one is the male, 10 years old, was admitted to the hospital mainly due to the sudden headache, vomiting, and coma for hours. Had CT shows the left hospitals the intracerebral hemorrhage and ruptured into the ventricular symptoms. The physical examination shows that the patient was less jerk and the meningeal aerations, the same was positive. Limbs can move as directed. This is the first head CT. From the CT, we can see there's the left hospitals the intracranial hemorrhage and ruptures into the ventricular. The patient to hospital complete the related examination. Intracerebral hemorrhage breaks into the ventriculars, which can easily cause the cerebral vascular and hydrocephalus. So on the second days of the admissions, we done the intracranial pressures monitor promise inflation and external the ventricular drainage. So after that is the CTA shows that we can see the AVMs in the brain. The post-operative the intracranial pressure was the normal on the 3 days. Respectively, the whole brain angiography was perfect still. I think that maybe after 5 days. Intraoperative finding, the left internal cardiac arteries angiography showed the branch of the upper trunk of the left middle cerebral artery supplied blood to the malformation vascular mass and drains to the tuberous sagittal sinuses. The bilateral vertebrae angiography shows the left posterior the cerebral arteries supplies blood to the malformation vascular through distal the branch and drains the tuberous sagittal sinuses. Yes, that's the DIC. There is the AVMs on the these others videos you can see there's the AVM. Yeah, we can see that the blood is the drainage to the central sinuses, central sinuses. Yes. After the results, and we got this operation for these patients. After opening surgery, we can see the left parietal accepts the vascular malformation. After it opens the duress, we use the fluorescence angiographies to advance the blood samplate arteries. Using this technique, we can see that the blooding the artery. So we slide. After that, the fluorescence angiography. From the videos, we can see when the arteries was blockade, and so the AVMs almost disappeared from the video. When we block the arteries, and so the operation is very easy. It's not and then the blood is very small. Yes, when the resection of the AVM composite and complete the resection rate. Post-operation CT scans, we can see that is the aerosol is normal, no bleeding. This is the case 2. This is the mouse, 14 years old, was admitted to the hospital mainly because of the sudden headache for 5 hours. This patient has the vomiting 3 times, accompanied by left limb weakness, unconsciousness. High CT shows right arteriosus hematomas ruptures into the subarachnoid space. Physical examination, the muscle strength of the left upper limb was grade 2. The muscle strength of the left lower limb was grade 1, and the muscle strength of the limb was grade 5, and the muscle tone was normal. It's the head CT. From the pictures, we can see the right parietal lobe and breaking into times. The surgical hemorrhage with the brain swelling. The CTA shows its AVMs here. After the cerebral and cerebral vascular embolization under the general anesthesias. Intraoperative finding are malformation vascular mass 3 centimeters to 2 centimeters in size can be seen at the top of the right side with the multiple branch of the right anterior cerebral artery and the distal branch of the right posterior cerebral artery is the supplying blood. Aneurysms are about 2 millimeters and 1.5 millimeters can be seen in the blood supplied branch of the right anterior cerebral artery. The malformation groups was drained by 2 cortical veins. From the videos, you can see the AVMs is very clear. This is the different direction. From these videos, we can see the supplying the blood, supplying the arteries, and the drainage veins is very clear. Post-operations, post-operations is the head CT. From CT, we can see there's the brain swelling is very deep, and there is the hematomas in brain. So we get the surgical treatment of the intracranial hematomas in vacation. So these patients, the first step is the embolization of the AVM. After that, and we using the surgical treatment to resection of the hematomas. Post-operative is conscious here, and his work where business his left arm left muscle strength was 2+. His left limb muscle was grade 2. His right limb the muscle joints and switch were normal. Yes, 8 months after surgery, and the patient come back to the hospital, and we got the DSA again. From this, you can see the AVMs is dispersed, and we can see the gloom. This other picture, other video. Next is the case 3. This one, the others patients males 15 years old. The main cause was the starting headache for 3 days. The patient suddenly develops the headache, nausea, and vomiting while studying. The brain CT shows the right temporal particles, the intracerebral hemorrhage. The physical condition is the jaundice, poor sprained normal muscles of the limb. Yeah, from the scan you can see the right temporal part of the intracoronary hemorrhage. So the cerebral angiographies, but the abnormal vasculars we're seeing is M2 and M3, the segments of the middle cerebral arteriole. Considering the average malformation and the drain veins, the rabbit vein, microaneurysms we're seeing is the abnormal. The visual masses is M2 and M3 segment of the middle cerebral artery. Yeah, this AVMs is not large, it's only the small. You can see there. This is the DSA. Choose the AVM. After that, we got this intracranial hematoma removes and arteriovenous malformation resection. Post-operative PET CT shows the patient was consciousness switch limb to the moment was normal. MRI of the height. Post-operative change of the intracranial hemorrhage, and the brain MRI shows the no abnormal. This others case 4 is the male, the 16 years old. The main cause is the headache with the disturbance of the consciousness for 7 hours. Cerebral headache accompanied by nausea and vomiting. Physical examination lack of the cooperation in physical examination, coma, that scores and E1VT M3. Inabilities to complete the muscle strength examination of limbs, increase the tone, and bilateral pathology scenes are positive. From the CT scan, there is the hematoma in frontal. The angiography, and from this cerebral angiography, we didn't see any AVM, but this patient only 16 years old. So we think here is the AVMs results the hemorrhage. After that, the surgical treatment intracoronal vascular disease, and intraoperatives, and we see there's our AVM. That's the AVM. It's not a normal surgery. After the operation, the operatives pass knowledge, hemorrhage and irregular vascular like structures were seen under the microscope of the left frontal lobe, consistent with the vascular malformation. After that is the CT scan post-operation. The hematomas is reversed, but in the ventriculars and sometimes the hemorrhage in here. The CTA is short and AVM is disappear. This case 5 is males 18 years old, mainly is due to the sudden conversions and loss of the consciousness for 2 hours. Generally, the conversions, the loss of consciousness of 5 minutes. Had the CT showed batched high distance the schedule and classifications in the left temporal superior lobe. From this, we can see there's the high distance mass here. So we think this is the AVM. This is the MRV. You can see it's enormous the winners in here. After the cortical branch of the left anterior and the middle cerebral anterior is the AVM, and drains into the cerebral sagittal and the left trans service the centers. Yeah, this is the picture of that. This shows the veins to the centers the intraoperative picture. You can see there's this the bleeding, this supplying that is the supplying the arterioles, and we block it. During operation, we're using the fluorescent angiography. You can see this is the supplying the arteries. So we block the edge after that and using the resection of the AVM. At first, the blocking the arteries, and so during the operation, most no bleeding is very safe, and we get a good results. Post-operative the CT shows that AVM is disappeared from here. And this patient, I know the disabilities, among their limb and speech and others function. Post-operative, the pathology is AVM with the thrombosis. The last case in the share is the male, 23 years old, mainly due to the sudden deafness and numbness of the limb for 1 day. The patient had a sudden deafness and numbness of the limb, accompanied by unsteady working, with autonosis and vomiting, and was conscious. Physical examinations, no overt abnormalities was found. The first scan we can see is the left arterioles outstated in hemorrhage, but the hemorrhage is not too much. So the patients, the clinics, the symptoms are similar. The MRI shows the vascular empty. After that, we're using the DSA. From the DSA, you can see there's a small AVM here. The supplying artery from the middle station, yes. But 3D identity, the sudden weakness of the limb, the cerebral meeting. So we got the emergency CT. The CT shows the bleeding again. So at the night, we got the imaging is the operation. Intracranial hematomas removes and resection of AVM. During the operations, now we can see as the AVM and pass the blood from the needle, and so we block the supply artery. After operations, the CT shows the AVM disappears, but some hemorrhage into the ventriculars. We can do the lung punctures and lung punctures drainage, and after that, the patient got a good recovery. So we have the conclusions about the adolescence. Brain AVMs in adolescence are often caused by hemorrhage, and the condition is urgent and needs to be dealt with in time. Surgical removal of hematoma and resection of AVMs are still the preferred treatment method. Pre-operative evaluation should be as comprehensive as possible. Examples, the CT, MRI, DST, and so forth. And if the intraoperative the fluorescence angiography is used in the operation, it will be more accurate, which can reduce the brain damage, reduce the bleeding, and improve the thrombotic effect. Make full use of the hybrid surgery. The advantage of the interventional surgery are also an important part of the treatment. That's all. Thank you, every professors, and I'm glad to have this chance and come here and to show our work in clinic. And if the shortage, I'm here and please discuss it. Thank you very much.
Thank you, Professor Jiang. Those were interesting cases. However, I have a feeling that there is no doubt that grade 1 and 2 AVMs are subject to surgery, or all of them with no exception in my mind. What do you do with unruptured AVMs? Dr. Shihan, did you hear me? You are muted. You are muted.
Please unmute here.
Yeah. I can see the professor. Yeah. If the unruptures the AVMs, and we think maybe if we have no the clinical systems, and maybe these patients that we didn't do anything about it. From the point of our study, we can see the average patients that get the symptoms in the clinic. At that stand, we show that is the others eruptors the trails that should, and maybe for the unruptured AVMs, any treatment is not good for this patient.
Thank you. You mentioned Aruba trial. I still believe that that is one of the most confusing results and most confusing trials which ever was performed, since actually there were 5 surgically treated patients in the whole trial. The follow-up was very short. In AVMs, you need to think about decades, not about single years. So actually, in my mind, the conclusion of the trial is do not treat AVMs by endovascular means. What do you think about Aruba?
Aruba. Yes, there are there's a robot trials that is following only the 33 months, and we think this time is short. Maybe just not maybe the 1 week ago, we get other patients in our department. That patients find the AVMs 10 years ago, but he got any treatment for it. But 1 week ago, and gets the hematomas in the brain, and so after that is a coma. So sometimes then we think maybe a long time the following up for the AVMs is very important. If this patient have any clinical symptoms, we think using the deal with it, that patients get the signals after the first findings of the AVM. Maybe 2 or 3 years ago, then the patients get the signals, but they didn't mention these symptoms and not to the hospital. But this time that you get this the hematomas this time get a not good result.
Okay. Are there any other questions or comments on AVMs?
We can invite comments. Professor Babak, would you like to ask something?
Yeah, so in a very interesting lecture. Thank you very much, Professor. I just want to know, first of all, how many of these patients annually your center operates on? And where do you see technology can actually improve what you're trying to do? So what are the limitations of current technologies, be it, design of a clip or kind of imaging? What are the technological barriers you see that you could mention? So 2 questions. How many patients? What are the technological barriers?
Okay, thank you, Professor. In our hospital, maybe with the only one centers of the medical, and we have the departmental work usually to do this the AVM. Maybe the no ruptures the AVMs and in the interveners others department. Our department is for the surgical for AVMs. Maybe 1 year we get the 50 to 100, but we have no the what is it? Then we have the we have established the numbers of it. 另外一个是就是问的是if the AVMs is not ruptures, and we think as we in the my point made, we think maybe do nothing is maybe the for the picture of the patients. But if there are sinners or others or other hemorrhage for this patient, and we think maybe the deal with the in time.
And in terms of the technological barriers, what are the shortfalls of the current technologies that you're dealing with?
So sorry, Professor?
What are the shortfalls of the current technologies that you're using? Being at even the surgery, what are the, what do you want to see be improved in the technologies that you're using in terms of the imaging, in terms of either coiling or clipping? Do you see things that can improve, and what are those things in the future?
as we know, and the interventional the treatment, maybe we're using the venous approach. From the venous stand to get the blood is blocking the AVMs. Others technologies, as we know, and in the surgical treatment, and we have no others technology, the new technology for this. Just know, and in our appointment, and we can see maybe the intra-operative ICG, maybe for these patients had important things for that. And we're using this technology, and after we're using this technology, and so every patients that are less bleeding and in the operation.
Thank you.
Raja, could I expand the discussion a little? Since we have Babak with us, and we are lucky about that. AVMs are something which is very clear in my mind. We have a series of 500 cases, and for me, it's obvious that grade 1 and 2 is surgery. No doubt about that, because there is nearly zero morbidity, mortality rate, and you cure the patient. Most patients are coming in their 30s. Grade 3 is a heterogeneous group where you can use all the modalities you have available. It means endovascular, radiosurgery, surgery, or observation. That's, as far as of now, easy. But grade 4 and 5 AVMs, this is a big, big problem, because anything you do has a higher morbidity, mortality rate than the natural course. You cannot beat the natural course in grade 4 and 5. So we have nothing to offer to the patients. We observe them, and 1 in 3 ultimately dies of bleeding. Do you foresee, with your insight into the technologies, any new modality or any improvement in recent modalities like staged radiosurgery or whatever, where which we could offer to the patients?
That's a question to me.
Grade 4 and 5 AVMs. Any new technology, any new modality, or improved recent modality? Anything?
So I would say potentially you could use nanotechnology, nanomedicine, do a targeted therapy, because when you're talking about grade 4, 5 AVM, you're talking about they're much more spread, and they're kind of difficult to either coil or clip. So how do you actually treat these patients? So I think some sort of I would say nano platforms that can redirect blood flow and basically literally coil or seal the areas that you could actually think that they will actually bleed, right? So not all of this will bleed. They're part of this AVM going to bleed. So if you redirect the blood flow and basically seal those areas that are in a higher percentage of bleeding, then you provide some sort of treatment. So there are, I mean, again, it has to be personalized. It's not going to work for everybody, but there are, a nano platform that potentially could be used.
As long as there remains one single shunt, as tiny as possible, the risk of bleeding is still the same. So some flow, you need to exclude the AVM from the circulation. If you touch the AVM and treat it only partly, there are published series that the risk is even higher after this.
So the point is, do not touch, but actually use NanoDrugs to actually seal the parts. So not necessarily a surgical approach. So much more like a targeted drug delivery.
causing, let's say, a myointimal hyperplasia to occlude the vessel, the similar action as the radiosurgery produces? Yeah. Dr. Xian, do you see something for grade 4 and 5 patients? How do you deal with them?
So, Professor, using this radiotherapy?
No, grade 4 and 5, Spencer Martin, grade 4 and 5, the most difficult AVMs, which we all subject to observation usually. What do you do with them? Do you treat them or do you observe them? Or?
Okay, if the if the grant is 4 or 5, and we think we at first then we use the endovascular interventions using this machine the method and to reduce the grade. After that, maybe the operation we can get it.
He's trying to downgrade the stage with multiple staged embolizations is what I understand.
Yes, embolization.
Right,
Yes.
thank you.
Dr. Xian, each embolization has a risk of 7%, according to the meta-analysis. So if you subject the patient to the risk to 2 embolizations, you subject him to the risk of 14% more serious morbidity mortality. The natural course is better than this.
Embolization, maybe if the patients have the ruptures and maybe the have the hemorrhage, and so most of the patients we get the operation for it. But just also and we see however others patient because there is the AVMs locate the petrous lobe, and so we using the endovascular stimulation and to embolize it at first. So after that, and we remove the hematomer from that.
Right.
Maybe this the best it's ours idea. Yeah. Thank you.
Thank you very much. I think we can take the concluding remarks from Professor Vladimir Benes and close this. Professor Benes.
Yeah, Dr. Xian, thanks for the great lecture. Those were very, very interesting cases, and you really did a good job. And I believe that everyone enjoyed the cases. So what I would say that the AVMs is a rare disease. Neither of us do see too many patients, and neither of us really is a big expert in this. Let's say Michael Lawton or those who are now retired. So thank you for the lecture, and Raja, thanks for organizing this big event, and the concluding words are up to you, Raja.
Thank you, Professor, and we learned many things from you and other professors. Thank you very much.
Thank you very much. So I'll close this officially on behalf of the Education Committee of the ACNS and the President, Professor Yoko Kato. I would like to thank both the speakers of today, Professor Babak Khatib as well as Professor Kian Rongjun, and the chairs, Professor Vladimir Benes and Professor Walter Jin. Thank you very much, my co-host Liu Bun Seng, for joining, and also Professor Shubin for broadcasting on the WeChat channel, and all the elite panelists who joined us. Thank you very much.